Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is September 25th, 2025, and we're talking to Daniel Dombeck. Dan is professor in the Department of Neurobiology at Northwestern University, and his work is on changes in brain activity during learning and performance of learned tasks, and he focuses on circuits in the hippocampus, entorhinal cortex, and striatum. He uses calcium imaging to analyze electrical signaling in awake animals that are performing learned tasks in a virtual reality environment, at least most of the time in a virtual reality environment. And I hope one of the things we can do today is learn something about these technical innovations that make this possible, as well as the content, the actual content in the research. So welcome, Dan.
Great, thanks for having me.
Also with us is Francesco Savelli. Francesco is our local hippocampus electrophysiologist.
Hello.
Francesco. And Stephen Jones, who works in Francesco's lab and who's also highly competent hippocampus guy.
Well, thank you, Charlie.
Yeah, we all vouch for him.
And me, I'm Charlie Wilson, your host. So I kind of like to start. I know we've had place cell podcasts before, and we usually jump right into that hard core stuff about place cells, but I don't think we've ever actually reviewed the idea of the cognitive map and why we think this is a compelling idea. We think we have one, we think animals have one, and we think that it has utility for them and that we can somehow find the part of the brain where that's done. So
yeah.
you could maybe collectively fill us in on that.
Sure. Yeah, I mean, I think the idea, the motivating question for our research, and that kind of leads to the cognitive map, is how we navigate in the world. How do we get around? How do we know where we are? How do we remember where we've been? And animals have to do that as well. They've got to pick a path to get from point A to point B in their environments and know where they are with respect to landmarks. And there's different ways that animals could do that. There's different ways to navigate around in space. Animals could be using just a sort of sensory response table that they've learned through their lives of what to do when they see certain things, or they could actually generate a map of their space. And there's reasons to think that map of space could add utility to survival for the animals. And so that's one of the main questions we're trying to address now, is why do we have maps? It looks like, with these place cells that animals have, grid cells that they have, those are the components of a map. But why did that develop? What was the driving force to develop a map inside their brains?
One of the amazing things about it is it's one thing, like for some psychologists like Tolman or somebody, to come up with behavioral reason to think that a rat learned a map of a maze or something like that.
Yeah.
It's another thing to find out where in the brain that's done.
Right.
And so the miracle, at least the one I watched happen, was people actually found a lot out about that.
Right. Yeah.
How do we know when we found the right part of the brain?
Well, that's a hard question to answer, I think. There's a lot of things that led John O'Keefe to the hippocampus. When he found place cells, right? When he found what we think are the cellular substrates of the cognitive map in the 1970s. And a lot of it came from Brenda Milner's work when she was studying epilepsy patients who had their medial temporal lobes or parts of them removed, and they had problems with memory, with remembering episodes of their experiences. And that really led researchers to then ask, well, what is it in the hippocampus or in these medial temporal lobe structures? What is it that's in there that's actually serving as the substrate that's helping them remember these experiences that they've had, right? And that's where people then went in. Richard Morris did water maze experiments showing that if you remove the hippocampus before learning or after there's deficits in the animal's ability to learn and remember where they've been. Kind of linking that hippocampus and memory to this ability that animals have, and it was then John O'Keefe went in and started sticking electrodes in and asking whether there's any cognitive map like cells, something that he could kind of map onto this idea. And I mean, he was well aware of the idea of the cognitive map. If you read his early papers, they sort of start out, they're sort of framed around the idea that there's this cognitive map, and that that idea would explain partly some of the things that Brenda Milner had seen. And then if you look at some of those early recordings, a lot of them we wouldn't actually call place cells today. This very first one is something more like a head direction cell, where they were kind of holding the rat and like rotating it around and showing it only fires in one direction. But just showing like something in the middle of the brain, like, kind of halfway between sensory input and motor output in this like cognitive area, finding something that has a correlation to like, a space to a behavior, something interpretable, was pretty remarkable. And I think that's, what captured everybody's attention.
So I'm always shocked because the cognitive map isn't the place in the brain that has the cognitive map isn't a map. I mean, you can't see the layout of the room.
Yeah.
It's like in the retina, there's kind of a map of visual space.
Yeah.
But in the hippocampus, there's no map in that sense of location.
That's right.
So
yeah, the visual cortex has a nice map, right? Orientation columns, for example. Yeah.
So what's it mean that the map is, I don't know, fragmented into cells that are distributed far?
Randomly distributed around. I mean, so I don't think anyone knows the answer, but I'll give you an idea. This is something I've been talking about for years. So if you have a system that it's meant to do the same thing over and over again. You have some neural circuit, and it's meant to encode the orientation of some visual stimulus, for example, and you have several neurons that are part of that computation. It actually makes sense to have them close to each other in physical space, right? You want to minimize the wiring distance. It minimizes, the amount of energy, it minimizes computation time. And that makes sense if you have a dedicated circuit to something that's going to happen a bunch of times. But if you have a system like the hippocampus, whose job it is to encode any arbitrary sequence of events that an animal experiences in its life, you don't really want to have a dedicated group of cells that are encoding, this room right now, for example, in my head, because then I'm going to need to use those cells again, tomorrow, next week, whatever, to encode something else. And so there's that idea of minimizing wiring and wiring distance between cells in a circuit is sort of counteracted by the need to maximize information storage capacity. And so that's what a lot of models, this is the idea that the hippocampus is really trying to maximize information storage capacity by maximizing connectivity between all of the cells, rather than having well-defined little groups that are kind of dedicated to something that they're going to do over and over again. That's the way I think about it.
So it's really more like a drawer full of maps, and you could pull one out and stick it on your hippocampus in whatever situation, like having a drawer full of topographic maps for hiking, and you say, I'm going this hike, I need this map. And so places on the map can't possibly be the same place in these different situations.
Exactly, right.
But the brain might have both, because it's possible that the grid cell system in input actually works more what you had in mind and be a universal map that is kind of nondescript and lays out everywhere.
Yeah.
And so works more like perhaps, the analogy of the visual system, minimizing the wiring. It depends on the model, because the jury is still out. We don't know how they come about. But according to some models, there would be that local connectivity and wiring, for example, the attractor network model. And so you had that. And then you have the hippocampus that somehow gets that, obviously it's a two-way street. It's not one-way street, but gets that and just creates this more erratic system that can have scrambling code in multiple environments. And it seems to add this possibility of creating unique representations of different environments, for example. And it might be very important for, contextualizing memories. So one thing about the first question you asked is, what's cognitive about the map? So you could talk about, you could have probably 100 people talking for years about that and not agree. But So one of the idea would be to be a component of a memory system that is heavily based on space. At least space is an important dimension of it. And given the hippocampus involved in memory, it kind of makes sense, for many years. But then there is this issue of maps. And so, to what extent the cognitive map as intended by O'Keefe, which is different from other lines of research and disciplines, is connected to our idea of a map. So the cognitive map of O'Keefe was the idea of a locale, so generally a surrounding, and not necessarily be the system, for example, that allows you to navigate complex mazes or routes. That could be a different system and a different part of the brain.
Oh.
So we had Doug Nitz here who did work in the posterior parietal cortex, and these route-centric that track the progression along particular routes.
Listener, go back and listen to the
yes, Doug Nitz in was that 2020? I don't remember.
That's a great point about grid cells, though, that they're sort of the in-between, right? But they do seem like they're organized, and if they're a metric of space in any situation, in any experience, space is space, and so it makes sense to have a dedicated circuit.
In an enclosure, but then the question is, if you're saying you're walking from, say, one room to another, or along a corridor, does that grid metric stays consistent metrically all throughout? And the evidence, or even for place cells, the evidence is it tends to kind of fragment into multiple maps. So it becomes more like a patchwork of maps that are not necessarily consistent within a single global reference frame that takes the whole city, the way that you see on Google Maps.
Yeah.
So there are multiple reference frames. And the question is, to what extent, an animal needs to really have a globally consistent map, spanning the entire, building or...
Yeah, probably not.
Whereas you can navigate around with a patchwork of maps, for example.
So does that work for us? I mean, I imagine I have a global map of my hometown. Is it really not a single global map of my hometown? I can kind of visualize it.
Well, one complication, though, in human navigation is that did you acquire that map all by yourself, or because at some point you actually looked at the map probably of your home.
Yeah,
so is it your visual system that is So it gets complicated with that thing. But there are different kinds of spaces, and so multi-compartment spaces are there is a lot of evidence from the psychology literature that people don't have consistent maps. People who have worked in hospitals for decades, and they know exactly how to go from anywhere to anywhere, you ask them to draw the map, they will swap things and make really metrical errors, because perhaps they use something that is more topological, to just navigate around. But then within the confines of those hallways or rooms or things, that's where probably the map of the place cells, grid cells, is being used.
Right, more in those local spaces.
Yes.
Yeah. Though there's some really interesting work from Nakamolinovsky in Israel, looking at bats flying over very large distances and carrying tetrode recording devices and actually finding place cells spanning kilometer scale regions. So I totally agree with everything you said about humans and most animals, but it probably also depends on what is the behavior that's being studied, right? And if we were out in the middle of the desert or something and hiking around, we might have large scale grid maps and place maps.
Yes,
and
that actually goes back to when I did my PhD, and I worked some with Benjamin Kuipers, that in 1977 introduced the cognitive map. So 1976 cognitive map, O'Keefe, and then Benjamin Kuipers, who worked with Marvin Minsky, just put this cognitive map. It was a completely different kind of cognitive map. And one distinction was exactly what you were saying. He distinguished between large-scale space and small-scale space, But large and small doesn't mean large and small physically.
Right.
It depends on your perceptual horizon. So when I talk about those things, I give the examples of a stadium, a baseball stadium. That would be small scale space because you can see the whole thing. It's a vista. It's just the whole thing fits your perceptual horizon. And then, you could have a large scale grid cell representation there. And then I give an example of one of the players going through the corridors of the locker room, which is physically ways. I mean, it's a small place inside. Well, that's large scale space. It's
right.
not according to the definition of Kuiper's, because now you got mazes, you got rooms, you can't see the whole thing, and so now it's a completely different cognitive process because you cannot acquire all the spatial information at once, modulo turning around. You have to piece it together with essentially integrating different sensory snapshots that you take over time. And so it's just a kind of a different kind of space. And so it goes back to what you're saying. It's like, if you're in the desert, that could, maybe the place grid cell system could be very relevant there. If you're living in an open space on a beach or whatever. But then if you're like in a room or a maze, then that could be very confined and fragmented.
Right, which is where most of our understanding comes from.
Yeah, exactly.
Yeah.
So there's a resolution issue. You only have so many place cells, and the resolution of your map must be somehow determined by that. And so when you say large scale or small scale, I'm imagining zooming in and asking about the accuracy of my statement that I'm in a particular set of coordinates on the map. And what's the information capacity of this? How much, like a map that's active at this moment, which I understand won't be active the next moment. Somehow other maps get stored in and can be brought into the hippocampus somehow. So, but for the one that's there right now, do we know anything about spatial resolution and the number of cells and just information problem?
Yeah, I mean,
we know quite a bit.
Yeah, so I mean, I'll refer back to this the bat work that Lenovsky is doing, some of which they just recently presented at SFN, looking at place fields from bats flying on this big island that they recorded them over, and these are bats that they've recorded in rooms like the size of rooms that we might have rats running in and recording place cells, and you see grid and place fields that are relatively small, pretty precise localization that you could do recording from 20, 30 of them. You could localize and decode the animal's location to a few centimeters. But if you take those bats and let them fly around in space, the place fields become much larger, and the resolution of those maps, reduces. And so you can, with the same number of cells, it's now mapping a much larger space, right? And so it has to do with what's the behavior and what's the resolution that the animal or the behavior requires, knowledge of location to be. If you're flying or traversing over a very large distance, your place fields can probably be quite big, and you don't have to encode at that fine resolution that you might in a room like this, where we want to know precisely where are we with respect to the chair and the table and things like that. So I think those behavioral demands really dictate the scale over which the cognitive map is.
So I read, like in this room, I have a map of space in this room. Does it take every hippocampal neuron to encode that? And so I could just say, what's the number of neurons, and divide that by the square feet of the room?
There's varying estimates of the fraction of cells that are place cells. It's a sort of somewhat accepted value is somewhere around a third of the hippocampal neurons are encoding any given environment that the animal is in.
So does that mean that's the maximum that could encode? Or what I was getting at is, do some maps require a lot more neurons and some fewer, and so you're not using all of them in any one moment?
Yeah, I mean, I'd say that that goes for pretty much any map, right? That it's about a third of the cells. If it's a continuous space, it's about a third of the cells that's going to be encoding that, maybe up to half or something like that. But there's, in a rodent, hundreds of thousands or close to 1 million of these cells, and there's a huge number of combinations picking out a third of those randomly, right? And that's where that information storage capacity comes in. that in every given environment, if you pick a random third, there's many different random thirds that you could use to uniquely encode every space.
So when you decode it, which I know you do because I heard you say so,
Yeah.
so you look at a bunch of activity of neurons in an animal that's moving around, and you can localize where the animal was at any one moment, so you know how to decode it.
That's right, yeah.
So I'm wondering about a part of the brain that has to decode that.
Yeah.
As sort of the next stage of processing or something, like you have access to a relatively small proportion of the total
Right.
hippocampal neurons at any one moment.
That's right.
And yet you do a good job of decoding it.
That's
So
right.
the next place maybe doesn't have to look at any
All
more
of
than
them.
the number that you are looking at.
That's right. I mean, it's a really interesting question of why in, say, a linear track that an animal's running in or an open space, why are there so many cells that encode the same location, right? If you have 100,000 cells, you take a third of them, and their place fields are 10 centimeters in diameter, and the space is a meter, you've got a lot of redundancy in encoding, right?
You could cover that with 40, 50 cells.
Exactly. So why are there so many that are actually there? And that it's an open question, right? I mean, some ideas are that most of those cells they're all sort of present and they're encoding the space at a very high density, and they're just sort of waiting to grab onto some thing that might happen at that moment, at that time. And so they're sort of encoding and ready to map that location to some experience if it becomes really salient, so they can like do single trial learning. And then if nothing really interesting happens there, those place fields sort of disappear the next time the animal is in that space. They weren't really needed, right? And so maybe it's better to over encode to start so that you can encode very densely and precisely if something, life threatening or, life important happens. That's one idea.
Yeah, that just goes back to the idea of what's cognitive about the map, right?
Yeah.
So if it's part of a memory system, you might need that redundancy to put more stuff in it. And so creating conjunctive representation with other things. Now, whether we need so many neurons, it's just hard to say. But because also it's hard to say, we have examples, but we don't have a lot of examples of other things attached to the map, that is other variables that are non-spatial. There is definitely evidence, but
a lot of people say that we remember episodes by creating a kind of spatial story.
Yeah, the what, where, and when.
And I mean, I guess I don't know what kind of evidence they're speaking from when they say that, but there's this sort of notion that the hippocampus encodes all kind of episodic memory by making fake spaces.
Yeah, well, it doesn't have to be physical space that's encoded, right? Today I was showing evidence of mapping olfactory spaces, and there's evidence of rats, for example, pressing a lever to change the sound frequency that they're hearing and trying to dial in a specific frequency to get a reward, and these place cells are mapping that sound frequency space, right? So it seems like any sort of experience that happens in a sequence over time can be encoded in the hippocampus, whether it's a spatial sequence or some sort of auditory temporal sequence can be stored through those same place cells. So that concept of what is a space can really be expanded to not just being physical spaces, but more abstract spaces or other dimension type of spaces that we can think about.
What an incredible challenge for the part of the brain that has to read this.
Yeah,
It's like, oh my gosh, it keeps changing its map spontaneously all the time, and it changes the scale of the map, and it even changes the modality of information that's being.
There are even multiple scales at the same time. I think we should mention that because there is a topographical organizational scale both in grid cells and place cells. So there are the same space is being represented on multiple scales simultaneously in the brain by both systems. And so there is also that, which might be useful, as you say, if you're on a big island, maybe the representation grows, but even if you're not in a big island, you might be interested in representing episodes that matter only to a specific location versus a larger location. And so there might be a matter of efficiency in how you record. So I would just kind of give you the examples to my students, undergrads. It's like I'm in the classroom, if I bump here and I hurt myself, like I was teaching, to the desk, want to just like pay attention to this here. But if it's you and you come and says, oh, this class is very boring or very exciting, that's about the whole thing that happens in the entire classroom. You don't want to have like 1,000 cells at a high resolution all connected to that memory. You might just want to represent it with just one big place cell, that one big place field of a place cell that covers the entire environment. I mean, it's a little bit of a simplistic, perhaps, way of thinking, but there might be something to it about that, just to kind of provide the intuition on why some of these things might be.
Agreed.
But I think one common theme that has been talked about in different ways in different times is this idea that the hippocampus, or in general, the whole hippocampal formation, has this ability to kind of produce an internal representation that is, and I'm not speaking very professionally perhaps, that is kind of eager to map and wrap around the external experience. So it's not, and so this is the idea of the brain inside out of Buzaghi and the book, but I mean, that idea has been present in various embryonal stages, during the history, because even the idea of the path integration is that, it's essentially this idea of predicting where you're going to be based on your self-motion and then reconciling it with the external information. So the idea has always been there. And then the sequence, this ability to create sequences both in space and time. So this is this idea that the hippocampus is somehow creating an internal representation that is looking for things out there to kind of hatch on. And maybe I'm just not explaining it. I'm just trying to explain it in an intuitive way.
I'm visualizing this sort of beast.
Yeah, exactly. So I'm trying to explain it in an intuitive way. And maybe I'm not sure I'm doing a fair job.
People often describe it as a predictive system as well, right? So it's, if you can build that internal model latching onto the space around you, one of the things that it could be useful for is to predict what's coming next, right? If I take a step in this direction, if you have the cells, there's ideas and data showing that these place cells are actually often kind of sweeping out ahead of the animal as it's moving, as if they're kind of maybe subconsciously predicting what's coming up, so that you're not just taking a next step and saying, oh, look, where am I now? And then the next, oh, look where I am now. You're constantly sort of anticipating what's coming next if you have this internal model of space or experience or whatever it is that it's latched onto.
It's like your model of the chessboard. After this move, where am I?
Yep.
Compared to the other people. So I don't want to be annoying.
No,
and I know that it's very
you
annoying.
never are.
It's annoying, and people do it so much that you say, I've discovered this about this part of the brain, and they immediately say, okay, well, how does the rest of the brain use that? What other part of the brain uses it, and how do they use it? And they just start asking you a bunch of impossible stuff as though you should know that.
Yeah.
But you've just made this huge discovery about this one part of the brain. You'd like to savor that for a minute, but no chance. So that's the question I'm going to ask. So what do we know about where in the brain the output of the place cells ends up and how it gets utilized? I know there's been a lot of work on how does a place cell get created from the inputs to the hippocampus, but how does a place cell map get used in the outputs? Vaguely, but where are the axons of the place cells going would be a first start.
Well, I mean, they go back. A lot of them go back to through subiculum, and kind of some back to entorhinal cortex. There's big loops that are present there. I mean, I think that's a good question, and a sort of parallel question that this brings to mind is the question of how useful are these place cells? How needed are they for navigation in general?
Oh yeah,
right. Like, I mean, so there's been certainly lesion studies where the whole hippocampus is taken out, and there's clearly behavioral deficits, as Brenda Milner and Morris saw. But what if you're talking about the specific place cells, what happens if you take out some place cells? Does it affect animal behavior? And there's a recent study just a couple of years ago, which I think is the first to really get at this question, where the researchers, this is Michael Heuser's lab in London, where they were able to do single cell optogenetics They imaged a population of cells, found where those cells fired in the environment, found their place fields, and then they could go in and activate individual cells. And they could activate all of the cells as the animal was running along the track. They could activate the cells that were active at the reward site, even though the animal wasn't actually there. And they actually showed that it changed the animal's behavior. The animal started licking at the wrong place because, the idea, the simplified idea is that they're making the animal think that it's over at that location, right? And so, that tells you that at least it isn't just some, representation, some, efference copy or something like that. It actually is used and can be used to drive behavior. And so those axons that are leading out of the hippocampus must go.
Somehow they do. We don't know what the next step in that process is.
I'd say it's messy. They project to a lot of regions, and those regions, there's a lot of these association areas. I
mean, we don't know so much about the brain. We don't need to apologize for that.
No.
I think it's a great question. Yeah.
I mean, related to memory, there was the index idea. So, as you said, it's all cortical stages, then go to entorhinal cortex, go to the hippocampus. This idea, the hippocampus is like the deeper part in which then things bounce back and go back up. Why? And so there is these ideas about memories, of creating indexes of memories that are distributed through many cortical areas, or the idea of memory consolidation. So the features that is reminiscent of episodic memory of the hippocampus is this immediate formation of place field, for example, or this idea of creating a memory right away. But then the question is whether you want to consolidate it or not. Consolidation might come from the repetition of that memory, for example, during the replay, during sleep. And so that could be what the hippocampus, one of the functions potentially that would be like to repeat. And so that systems in the cortical areas that learn slowly can benefit.
As somebody who works on other parts of the brain, I have the impression that the hippocampus investigators' view of the brain is a little bit like the New Yorker's map of the United States. It's like, here's the hippocampus, and then there's all of that other stuff. So, and that's what I just thought. I heard you say.
It is at the center of the brain.
It is at the
mouse
not the thalamus. It's at the center of the brain.
Well, so there are actually place cells in the visual cortex.
Uh-huh.
So as animals run down linear tracks, after the experience is no longer novel and it becomes familiar to the animal and they've formed a memory of this experience, there are cells in the visual cortex that look like they're encoding location along this visual track that are not just responding to only visual features.
And
so
they might be informed by the hippocampus about
that's
that.
right. This sort of gets to this indexing idea. The hippocampus is the first to encode these sequences and this experience, and then it offloads those memories to cortical regions that actually store them, and then it just sort of becomes this system that is the index that says, okay, that's where the memory of that experience was. But
cool. And
that direct connection, is complicated. It's a good question.
I don't mean to.
No, it's
not
it's
annoying.
a good question.
Yeah.
There are some ideas.
When we don't know the answer, it's a good question.
So I want to change the subject because I can't let you get away without asking you about the fact that you do all this work on hippocampus. Your lab is very productive. I looked over all your recent papers. It's like a busy place with a lot of interesting hippocampus stuff going on. But then there's also this whole other layer of what you do that has to do with dopamine and striatum,
Parkinson's disease.
So I think it's kind of unusual for people to be doing two unrelated things like that, although we've had other people on our podcast that are like powerhouses that do that.
Yeah.
But those two seem really...
Yeah. And I've tried for years to merge them back together. But
so how did that happen, and what keeps it going?
So the reason this all came about from I had a postdoc, Mark Howe is his name, who came from Anne Grabiel's lab at MIT, basal ganglia lab. He was interested in dopamine, and he came to my lab, and this was early in my lab's history, and I was all completely focused on place cells in the hippocampus, and he came and he said, well, if we image in the basal ganglia, there'd be all these wonderful things we could do. And I'd say, yeah, but look at the hippocampus, look how great this is. And for, a year or so, I got him focused on the hippocampus. And, but then, I'd talk to him and I'd realize that he had actually kind of, gone back and had ideas in the basal ganglia and was playing around with windows over there. And I'd kind of reorient him back to the hippocampus. And one of the projects he was supposed to work on was to look at how to record neuromodulation in the hippocampus, and to look at, say, dopamine axons from VTA to hippocampus and see how they're active when animals are forming memories. And so he started doing that experiment. And then he said, we don't really know what dopamine is supposed to do in the hippocampus. He said, why don't we go somewhere where it's really well known what dopamine does? And so he finally convinced me that, okay, fine, put your window over the basal ganglia, label dopamine neurons with GCamp, look at their axons, and let's confirm that the method works. This was sort of early days in the optical recording of these cells. So, go and find those reward responses that are supposed to be there. And so he put his window in, imaged these dopamine axons, and he couldn't find any axons that were reward responsive. And so we were like, well, what is this? This doesn't make sense. You give the animal a reward. It's an unexpected reward. It should lead to a signal. And instead, what he was seeing was that as the animals are running on the treadmill, there's this pulsing activity in the dopamine neurons. And I mean, there were sparse descriptions of motor responsive neurons that Schultz and others had recorded in primates, but very few of them. And the whole field had really kind of switched to the idea that all dopamine neurons encode reward prediction error. And it was really kind of confusing and convoluted how that is related to movement, because if those neurons die, people get Parkinson's disease. And here what we had found was this whole population of dopamine axons that didn't care about reward and only cared, were only active when the animal was running. And so we couldn't ignore that, right? I mean, that really led to this whole area of research in my lab. We, published that paper. We found that there's actually, across the striatum, there's some dopamine neurons that project to this more dorsal region that are encoding movement and not reward. And then as you go more ventral into the accumbens region, you find the more classical reward prediction error cells. And that was, I'd say, I think a pretty major finding. And it kind of, helped us understand how losing dopamine neurons can lead to movement deficits. If they're just encoding rewards, that's complicated. But now if there's this population encoding movement, that makes more sense. As we did more work, we found that the cell bodies of those neurons that were encoding movement actually sit in the ventral tier of the substantia nigra. That's the first place dopamine neurons start to degrade in Parkinson's disease. And so then that connection really led us to collaborations with people at the medical school, looking at gene expression patterns of those dopamine neurons, and really trying to connect to Parkinson's disease. So it really just that one, sort of accidental discovery of moving the window over, using a new technique that nobody had used before to image in a region and image a subtype, cell type that no one had imaged there before, led us to find these dopamine neurons that we think had just been missed for years. And that's what led to that whole area of
persistence
research.
of a trainee.
That's right. I thank him every time I see him. I think. Yeah. And so, I credit him with that persistence.
Paying off.
It is. Yeah. This has really led to, we have a five lab team funded by the Aligning Science Across Parkinson's Foundation that's basically funding this idea of different subtypes of dopamine neurons that are not just functionally different, but they have different gene expression patterns, and trying to determine how that's relevant for Parkinson's disease. What is the gene expression pattern of the most vulnerable cells? How can we target those cells? How can we make them, less vulnerable? And it's really been exciting.
Love to have you come back and just talk about dopamine.
Yeah, that's a whole separate. That's a whole other hour talk. Yeah.
But as for now, I think we are out of time. So thank you, Dan. It was really great.
It's fun to be here. Thanks for having me.
And Francesco and Stephen. This has been Neuroscientist Talk Shop.